Okay. Is it week 6? Oh, I think this is actually passed over wrongly. It's probably week 5. No, it is week 6. We're doing an introduction to neurophysiology. We're now leaving the realms, well, I will leave the realms of the cochlea, the primary sense where the vibrations are changed from mechanical to electrical. That's what we've talked so far. And now we will enter the auditory nerve and the structure that lies beyond that, the auditory midbrain, the auditory brainstem, and then the auditory cortex. These are all fairly, this is a very basic lecture, which if any of you have a background in biology on a undergrad level, you'll probably find that very straightforward. But I decided to put it in here because I think it's incredibly important to understand the basics before we actually try to understand all the functions of the brain, specifically of the brainstem, where we will spend some time to try to understand how the neurons in the brainstem actually code the sound. This is interesting for two reasons. Firstly, because it's just interesting to understand how the brain codes sounds and how we actually hear it, and we've learned a great deal about that in the last 10 years or so. It is interesting because we can apply it in certain measures, for example for implants, for cochlear implants, but also more directly for brainstem implants. And thirdly, we can learn a lot about the functions and the missing functions when something goes wrong, specifically in the aging brain, and also learning, and all of these things are very closely related to the basic neurophysiology. So what I'm going to talk about today is the neuron. I want the learning outcome today. I want you to understand how a neuron works, basically, not in all details. That would be taking a whole degree probably. But for our purposes, how it works means how it responds to sound in the auditory nerve. So sound comes into the brain. We've talked about that for the last 5 weeks. And now we find out how it is actually represented in the auditory nerve, and it goes into the brain. So we'll talk about how they are activated, about threshold, synapses, about action potentials. That's what it's all about, because that's what we actually measure usually. We talk about how neurons translate the electrical signals from the inner hair cells into chemical signals and back again. We talk about excitatory, inhibitory neurotransmitter, integrating signals, and very just one slide about computation, but we talk about that a little bit more in the next weeks. Okay, here's a box standard neuron out of a textbook. This in fact is a, well, it is a box standard. You find that in the cortex, but you also find that actually in the cochlear nucleus. That would be classified as a bushy cell because of its bushy appearance of all the dendrites. So what do we have in an axon? In a neuron? We've got the dendrites, we've got the cell body, we've got what's called an axon hillock, and we've got the axon. The axon and all the dendrites look pretty much the same, but they're not. They're very much different. Namely, as we see in a minute, the dendrites act as integrator, they act as information collector, and the axon is the actual thing that does the computing in a neuron. That presents all the information from the neuron to further stations down the line. Now if we do a very simple experiment, thought experiment, but that's actually not too far away from the truth, we can do that. We stick a little electrode, a recording electrode, into the neuron. Try not to hit the soma. And put another electrode in there as well, in which we inject a little bit of a current. So we artificially change the membrane potential. This is what it's all about. The membrane potential is actually measured here. Remember the membrane potentials of the inner and outer hair cells? What was it for the inner hair cells specifically? Never forget these numbers. -70 for the outer, -45 for the inner hair cells. That's right. -45 to -70 is the general range. If you look here in the recording, is at -65, which is pretty much average, box standard average for any cell in the human body, including neurons. So it's slightly negative. It's actually not slightly. It's actually quite a lot negative. There's a lot of energy spent on holding it the membrane potential negative, which means if we now inject a current, and there are pluses in here, so we actually put a positive current into it, we depolarize the neuron. That's what happens. We depolarize the neuron here when we put a little positive current in there. Okay? Any kind of standard linear system would respond in a way that the membrane potential then goes up. But what the neuron does instead, it changes into a pattern. It changes its membrane potential into these very characteristic responses, which are called action potentials or spikes. I always call them spikes. I always slip, even if I try to call them action potentials. Subconsciously, I slide into spikes. The reason is for that. If you do an experiment in a lab, you usually attach this measurement to a loudspeaker. You listen to it. Why? Because you hear what's going on. It's all on a time scale that is very intelligible for us to follow what's going on. And then each of these is a click. You hear click, 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 click. Or in fact, it goes more like grrrr. And that follows speech, for example, quite faithfully. It follows sound. Well, if you have an auditory neuron, if you have a visual neuron, it follows light. If you send light to the brain, they will respond to that. But you can hear it. That's what I mean, and that's why we call that spikes quite often. Okay. Right. This is called an input depolarization, and the potential that actually generates one of these spikes is called a generator potential. And that's obviously closely related to a threshold. Okay, let's analyze that. We have two features here. Most important feature is how much input current do we actually put in. This is an artificial situation. We put artificially current in. Later on, the neuron does a very similar thing itself, but less controlled via the synapses. And we will see that how the synapses change the membrane potential from the outside, but we do it from the inside in order to do a controlled experiment. So here we, over time, we inject three different currents: a small one, larger one, very large one. And you see what happens if they in the other electrode, where we record, namely a change of the membrane potential, but basically nothing. And here, few spikes, and then a lot of spikes. So obviously, there's somewhere around here, there's a kind of threshold. So if we go above the threshold for the depolarization, we generate an action potential. And when it goes even higher, we do not increase the size, as you might think of these action potentials, but we change the frequency. And that's literally the frequency as in spikes per second. So if you're technically inclined, what that is a basic AD conversion. We have an analog signal that goes in, and we have digital signals that come out, namely a series of zeros and ones. 0 means no spike, 1 means spike. Here we've got 1, 2, 3, 4, 5 of these spikes. Here we've got 10 of these. And if we knew all of that, we could say, if we measure 5 spikes, we probably have that input current. That's a simple calculation. Okay, let's zoom into one of these action potentials. First, the axis, as always. I put on here roughly 5 milliseconds. That really roughly, because it varies across the brain, across the neural system. We're not only talking of neurons in the brain. You find neurons everywhere. Yes, you find them in every nerve specifically, and obviously in the well, mostly in the brain, obviously. But we do find them in all sorts of fashions, but they all share the characteristics of these action potential of this shape, namely the rising phase, the overshoot, falling phase, undershoot. Just the timing is different. Specifically in the auditory system, we work in the fastest system in the brain, to our knowledge, because the auditory system is so much tuned to calculate temporal cues that it needs to be very sharp in time. And so these 5 milliseconds are usually more like 1 millisecond in the brain, in the auditory brain. So it's all over in 1 millisecond. So you can, in theory, have 1,000 of these per second, in theory. In other systems, like in the motor system, these 5 milliseconds is a more reasonable assumption. 5 milliseconds for a muscle, for example, or for the vision, is a long time. Sorry, it's a short time. It doesn't matter. 5 milliseconds is long compared to the auditory system, the auditory signal, but not to anything else. Okay, so on the y-axis we've got 0 millivolt. That means nothing, death. There's a hole in the membrane. Then you've got 0 millivolts, and usually the resting potential around 65, 70 millivolts. Now we've got a depolarization. When it crosses the threshold, which isn't in here, this is like around here, then the action potential takes off. It goes actually positive for a few moments here. Then it falls again. It goes below the resting potentials for a while, and then it recovers. Okay. The repolarization. This is a depolarization because it depolarizes. It goes towards 0. That's the definition of depolarizing. Polarized means away from 0. Depolarized towards 0 and repolarized back again from 0. There's no sign in that. So depolarization can come from top or from the bottom. Doesn't matter. From our case, it's from -70 to 0, overshoot, and then repolarization. This thing here, so that when it crosses the membrane, the resting potential, and goes back, is an important time, because in this time it is either impossible or very difficult for the neuron to respond again. That has a name. That's called the refractory period, from here to here. And there's a bit hand waving here because there are a number of these refractory periods. There's an absolute refractory period when the neuron can't fire again, which is literally actually the time from here to here. And then there is a relative refractory period when it is unlikely that it fires again. And that can carry on for a number more milliseconds. And these refractory periods also increase over time with stimulation. So to give an example, in the auditory system, I said in theory we could achieve 1,000 of these spikes per second in a neuron. In practice we can achieve about 200, maybe 300 for a very short moment in time, maybe a few hundred milliseconds, and then it decreases up to maybe resting of 100 spikes per second. But still, 100 spikes per second per neuron, or maybe even 150, is perfectly reasonable. Yes, that can happen. That's what results in adaptation. So the neuron will wear out. We'll see that later on, what that actually means. But the neuron initially is very susceptible to stimulation, and then it will gradually stop being sensitive. And one of the consequences is that the refractory period increases over time. Okay. The action potential, as a definition, is an all or one change in voltage. It can never go, going back, it can never go halfway up. It can only go up to the top and down again, or nothing. It naturally occurs at the axon hillock. We'll find out later why. They do not occur anywhere else in the body, or not even elsewhere in the neuron, not in the dendrite and not in the cell bodies. And there is a very nice animation here that I recommend if you want to get a bit more detail about that, where you actually play around and push the potassium and sodium ions back and forth, and you can do it all in a playful manner here, but we'll see in a picture in a minute. The interesting question, this lecture always allows me to philosophize about why things are how they are. Why do you think action potentials are a good way of transmitting information? Or as a challenge, why not using graded potentials, like more input, here more input, higher action potentials? Why has nature gone the way of defining the frequency of these action potentials as the information and not their height? Interesting question. I don't claim that I can give a full answer to that, but there are a number of obvious hints towards that. And one of them is that every animal in the animal kingdom has exactly the same action potentials. Talking about fruit flies to what are the most primitive neural systems? It's probably no, not in fungi, they don't have it, but in, what are they called? The things living underwater. look like a bath. What? Crustaceans are quite advanced, actually. I mean, they're no, much more primitive. Sponges, SpongeBob. That's probably the most primitive animal in the animal kingdom, one of them. And they have a neural system. I don't know how many. They've got about 50 neurons, probably. As does the fruit fly, by the way. You know the fruit fly, the standard genetic test animal for every lab in the world. That has exactly, correct me if I'm wrong, something like 120 neurons that are all named and labeled, and they all have exactly the same neurons. It's not very complicated, obviously. Well, it is actually quite complicated, but it's not very excessive. It's just really you can label every neuron in every fruit fly because they're all the same. You can do fantastic experiment with them because you can take one out, or you can genetic experiment. You can have little changes where you actually just have half of the neurons or something and see what happens. This is why we learned quite a lot about that. But they all share the same action potential, the same chemicals, the same action potentials, and the same coding mechanism. And my answer why that is the hand waving answer, why that is so useful, is because it doesn't change over distance. And scaling is something that's incredibly important in nature. As you know, it doesn't really make a difference if you have a small animal or a large animal. Usually the same small animal grows to larger size. They have slightly different physical properties then, and slightly different problems to encounter. But the one thing that changes, that doesn't change, is that their neural system still remains the same. And the biggest animal on the planet is the blue whale. It's my favorite example. If you imagine a blue whale 35 meters long, it must have neurons that run from the fin of the tail up to the brain, 35 meters across the whole animal. And how would a signal look like if it is not coded in that way? And this is the reason, I think, why the action potentials are so powerful, because they can be transmitted over vast areas, vast distances, without changing form, because they're self-stabilizing, as we'll see in a minute. They look always the same. They're very simple to generate. They travel quite fast, and they never change shape. Whereas if you use the shape to transform information, you have a problem, because then you need to be careful to actually not change the shape. Here you don't have to worry about that. Okay, here is how it's generated finally. We need two kind of different chemicals for that, sodium and potassium. In fact, we need a few more, chloride, other anions and magnesium and some others, but this captures 95% of the dynamic of a standard neuron. Namely, we start at the resting potential here. Here's the threshold, actually. This white line goes up to number 2. This is a depolarization. Then number 3, rising phase. Number 4, falling phase. 5, undershoot. And then we go back to the resting potentials. Where does it start? Here, number 1. We have the cell membrane. Okay, it all happens, good morning, it all happens in the cell membrane. And we have channels, and we have a lot of channels. We've got millions of channels per square inch, square millimeter. But here important for us are sodium channel and potassium channel. And we have another channel that we have on a few slides later. But obviously sodium channel only works for sodium, potassium channel only works for potassium. Now we have a large negative resting potential, minus 70 millivolts. Where does it come from? It comes from basically the fact that the cell pumps out sodium. Haven't talked about that yet, maybe here. This is the sodium pump. Maybe I show that first. This is what the neuron does all the time. It takes sodiums, these Na pluses here, and they are inside and outside of the cell usually. That's standard. They're just floating around there. But the cell itself takes ATP, so the general unit of currency of energy in the body. It packs 3 of these sodiums into the channel, translates one of the ATPs in an ADP. That one phosphorus is left over. That needs to be resolved. And for it, 2 potassiums go into the cell. Now this is happening all the time. It's called the sodium-potassium pump. it uses 2 ATP to pump 3 sodium out and 2 potassium in. What happens over time, if we do that a lot of times, we build up more pluses on the outside than on the inside. Or in other words, a slight negative potential on the inside. This sodium-potassium pump is completely ubiquitous, again, across the animal kingdom. And not only in nerve cells, every cell in the body does that, and it is estimated that something like 30–40% of body energy is spent on pumping sodium out of cells. So imagine if you eat your lunch later, maybe not half, but a fair bit of what you eat goes into this sodium-potassium pump. This is the single most source of energy. Certainly in the brain, where it's incredibly important, but every cell does that. Right. So we have, that's what these pluses mean here. We've got all the pluses on the outside. We don't actually have negatives on the inside, but this is just to charge the balance. There need always be a balanced charge. It should indicate that this is negatively charged. We have more pluses on the outside than on the inside. Okay. Now we have an event. Either we put in current with an electrode, or we have a synaptic event, which is now here number 2. And what happens is that the sodium gates open. It's called activation gate. And this event, whatever it does, it opens sodium gates. It doesn't do anything with potassium gates, but the sodium flows in. Okay, this gives a little bit of a change in there, but it only happens to the time when the sodium is actually in balance inside and outside. But then something else happens, namely in 3. No, sorry, what am I saying? A little bit of sodium changes and activates the gates so that the rest of the sodium flows in. Sorry, the equilibrium of sodium happens at the top here. This is when no more sodium can flow into the neuron. So this is what's called a catastrophic effect. So some of the sodiums open more of the sodium gates, and all of the sodium from the outside rushes in and destroys restores the equilibrium up to the point where nothing happens there anymore. At this stage, they close. It's called an inactivation gate. The sodium doesn't matter anymore, but it needs to close so that it will be able to restore itself. And at this stage, the potassium channels open. Now there is not so much. There's actually a little bit of a positive overshoot now. So it is more positive on the inside than the outside, because we're above 0. This is actually a bit confusing here. And so the potassium wants to go out, but the electric pump here, or the electric pressure, is not so high as the chemical, because we've got a lot of potassium built up on the inside that needs to go out. So we've got 2 effects, namely the pressure from the electrical system and the pressure from the chemical system. You know that if you have a lot of sodiums on one side, then it can be even in equilibrium if you have it with potassium on the outside, but there's a chemical gradient that pushes both back and forth. So that's the reason why the potassium wants to go out. So all of the potassium channels open and being pumped out up to the resting potentials, where we up to the undershoot underneath the resting potentials, where it's all then in equilibrium again, but all the channels close, and the sodium-potassium pump over time restores then the resting potential. Because we're faced with the wrong situation, because we now have all the sodiums on the inside. They're left from here. The sodiums are all gone to the inside, and now all the potassiums have gone to the outside, and that's exactly the opposite way that we started. So now we need a little bit of time, the refractory period, in which the sodium-potassium pump starts pumping it back out again. That's it, in a nutshell. The action potential has been discovered by 2 guys in Cambridge. At least yes, Hodgkin Huxley, you heard of them? Only Nobel Prize for, not the only, the first Nobel Prize for neuroscience. They have used squids as their experimental animal and pushed electrodes inside. Do you know why squids? Because they are readily available. I could go down to the market and buy them. But also because squids are a certain kind of animal that are evolutionary on a different branch. They haven't developed myelinization, which I haven't talked yet. Which means, say that in a second, which means that their axons are really, really large. They are actually visible by naked eyes, about 1 millimeter diameter, and you can actually stick an electrode. You don't even need a big microscope for that. You can stick electrodes into the squid's axon here, and you can put a current in here, and you can measure what happens there. And that way, they put all sorts of chemicals on inside and outside of the neurons, and they came up with a set of Hodgkin-Huxley equations to explain that, or to model that. And they have, in the 50s, established this system. Okay, resting potential. I mentioned two inside sodium, inside potassium, outside sodium. These are the biggest one here. This is the axon here, sorry. This is the neuron. The axon branches out into the dendrites here again. Blown up. Here we have the membrane itself. We've got the pluses on the outside and the negatives on the inside. So we've got this minus 65 milliwatt resting potential. The main component of that is the difference. We have lots of positives here as well, namely the potassiums, but we have even more. We've got a few number of chlorides in here, but we also have these anions, which are just a collection of a lot of more complex chemicals that are negatively charged. But on the outside, we've got this sodium, 150 millimolar. That makes the main contribution to that. And the chloride as well, which we haven't talked about yet. But that doesn't play a role in the action potential, and therefore I'm very short on that. Okay, here's the sodium-potassium pump. That's building up the potential. And conduction in an axon. We've got now we have the action potential. Now we want to send it in the blue whale from the tail to the brain, 30 meters. How does that work? And we were talking in the human here. The distance from there to there is about a micrometer, a millionth of a meter, and 35 meters long. You can imagine that's a slight. How does a neuron find the place where it projects to? That's amazing. Anyway, now what happens? We've got the sodium and the potassium on the outside. The point is that we have all of the sodium, potassium pumps all along the axon, completely filled with it. And if we have a positive charge coming from here, or an action potential coming from there, it will raise the membrane potential at this stage and swap it around there. That in itself will shut the gates and will propagate the positive charge to the right, from the left to the right. So it's a chain reaction from the left to right. You have first you start with a negative inside. The sodiums are pumped to the inside. Potassium goes out. That changes the membrane potential. This is the area where the action potential happens. And here nothing can happen because you're on the left. Because you're in the resting, you have actually too much sodium inside, too much potassium outside. So you're in the refractory period. So it can never go back. It can only go forward because here you're still freshly charged, and the gates here will change their state depending on the membrane potential. They will sense the change in the membrane potential, will open, and so will carry on. It will basically jump from here to here to here to here to here to here and so on. And that goes very fast. How fast? What do you reckon? Kilometer per second? Speed of light? Speed of sound? How much is the speed of sound in air? 340 what? Good. What's the speed of light? .300 kilometers per second. Anyone else? 300,000 kilometers per second. That's right. So this is electricity here. That actually changes, by definition, with the speed of light. Is that as fast as it goes? No, it's not that fast. It's actually a chemical reaction, which is not that fast. Chemical reactions are the speed of chemical reaction are directly proportional to the size of the involved distances, because all of this is the pumping and the it's diffusion processes, and diffusion goes very, very fast on a short time scale, but very slow on a large time scale. That's why all of these things only work on a short time scale, not on a short time scale, on a short scale, meter scale. That's why it only works if you have that on in the area of micrometers. That fast, at least. But there's tricks. So, still, is that? Let's give you three numbers. Is that 10 centimeters per second, 1 meter per second, or 10 meter per second? $10 meters per second? It's not that fast. It's about 10 centimeters per second, actually. This is actually, because this is the standard, the primitive way. It has to jump from each gate to the next, which is not that fast. 10 centimeters is not for an insect. Is actually quite appropriate. There are also tricks, as I said, to make that faster. And the one trick to make it faster is actually to make it bigger. Why? Because you fit more gates, more channels on the surface. That's why squid do it. They do have to pack more channels on the outside, and that's why they make it bigger. Humans have gone a different way, or in fact, mammalians? No, who was that actually? Vertebrates. Squids are no vertebrates. Vertebrates have gone a different way. They have invented myelinization. Now, myelinization is swan cells, which are white cells generally, which clutch around the axon, and only the axon, nowhere else. And each of these is one cell, and they go all along the axon, at least in all myelinated matter. Have you heard of the term grey and white matter in the brain? This is where it comes from. The white matter is all the stuff that is actually myelinated, because this is connection. These are not neurons, these are axon. The grey matter is the neurons. There are lots of areas in the brain that are not myelinated because they are neurons, neural bodies, and only the axons, the wires if you like, are myelinated. The auditory nerve, you'd be not surprised to hear, is obviously myelinated. And why? Because it goes faster. Why does it go faster? Because it insulates the axon. You would think insulation is not a good thing, but in fact it is, because in between the axons, you have in between the Schwann cells, you have a little bit of a gap, and only here the membrane potential can change. This is the only place where you've got the what's it called, a lot packed of gates, of sodium and potassium gates, and if you change the membrane potential at one point here, then the excitation will jump to this point with effectively the speed of light. So you have a change here, a slow change, but it will jump to the next one, and then it will jump to the next one. So it goes jump, 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 jump. It's saltatory, it's called. It's Latin for jumping conduction. And that speeds it up by a factor of 10 or 20 easily. That way you get to 10 meters per second, in fact. Still, if you kick the blue whale against the tail, how long does it take for it to realize that it was kicked? 3 seconds. If somebody steps on your toe, it takes you easily half a second or something before you actually realize it. And one of the most fascinating things about the brain is that the brain constantly lies to you and makes you believe that everything happens at exactly the right time, although it takes in fact a real long time for you to notice that something happens. but your brain doesn't tell you that it takes that long time. It was fascinating, but a different lecture altogether. Okay, now how do these signals actually jump from one neuron to the other, or in fact from the inner hair cell to the auditory nerve? Through synapses that translate the signals. And synapses are quite complex and at the heart of a lot of research, because this is where neuropharmacology is at home. And I only describe the ones that are relevant for hearing, because the hearing process in itself is quite encapsulated, if you like, in the brain, and only makes use of a handful of different kinds of neurotransmitters, which are different from other neurotransmitters that are used in the brain. The ones that we are talking about are calcium activated. We've got the nerve impulse. Imagine this is the auditory nerve now, which projects onto some neuron into your brain. It would sit in the cochlear nucleus. We'll talk about that later. But this is where the auditory signal now actually comes from. So the action potential travels down from the axon, which sits somewhere much higher, until it reaches a terminal. This thing is called terminal, and there's an actual gap. So here it can't carry on, and all the channels, the calcium are still there. The sodium and potassium channels stop around here, and are replaced by something else, namely calcium channels. So the depolarization opens calcium channels here, number one. They still work exactly the same as sodium channels. They are voltage activated, so the voltage changes, but instead of sodium now calcium flows in. The calcium itself does something chemical, namely it causes the vesicles in the synapse to fuse with the the presynaptic membrane. So in the synapse, what the synapse does all the time, it produces neurotransmitter. There's a chemical, sometimes a simple chemical, sometimes a more complex chemical, but this is generated by proteins that live in the synapse, and they float around in there in little vesicles, and these vesicles are encapsulated with the same membrane than the neuron itself, or the axon. And the calcium makes these vesicles fuse with the presynaptic membrane down here, and so the neurotransmitter is released into what's called the synaptic cleft, so into nothingness. Okay, this is all the presynaptic process, because at this stage nothing really has much happened apart from that the action potential is translated into some neurotransmitters. These are green here, obviously, released into the void. What happens with that is totally up to the postsynaptic neuron, which is that one underneath. Postsynaptic membrane that does something with this information. First of all, the distance of the size of this postsynaptic, the synaptic cleft, is absolutely tiny, because this is not an electric process. Obviously, this is a chemical process. So we're talking about diffusion. As I said, the diffusion coefficients here are really slow on a larger scale. So we need actually in the area of nanometers between the two cells. So that is really fast. They can float around in there, but it doesn't take them very long to go from the presynaptic to the postsynaptic membrane. Now, as I said, on the postsynaptic membrane sit other ion channel, but they completely depend on the system now, which ones they are, and it depends on what they are doing with the chemical that is released, so with the neurotransmitter. In this case, or actually this is quite always the case, the neurotransmitter that is released binds with the ion channel and does something. It's not just a gate that opens, but they are quite selective sometimes for what actually happens. Okay, here's a list of the neurotransmitters that are relevant in hearing, and we have basically one excitatory, one inhibitory neurotransmitter. Talk about what that means. The transmitter can do something to the membrane potential postsynaptically. namely either raise it, so depolarize it, or decrease it, repolarize it, or polarize it. And you can do both, but it depends on the neurotransmitter which one is which. So you have a synapse can either be positive or negative, or excitatory or inhibitory, and it either leads to an excitatory postsynaptic potential, it's called EPSP, excitatory postsynaptic potential, or inhibitory postsynaptic potential. So whether a transmitter is excitatory or inhibitory depends totally on the receptor. The presynaptic neuron doesn't know that, it just throws it out, but the postsynaptic neuron takes that and well, uses it the way that it wants, either positive or negative. But if a synapse is either positive or negative, once it's determined, it is forever. And we have, in fact, the most important one, the most important neurotransmitter for us are acetylcholine, which is excitatory. It's the same neurotransmitter in vertebrate skeletal muscles. And in the auditory system is always excitatory, as in muscles. And in the auditory system, the most important inhibitory neurotransmitter is GABA, gamma-aminobutyric acid. We have sometimes glycine in higher ones, glutamate in other brain areas, but GABA is the one that is 80–90% used in auditory systems. Okay. There will be a test on the chemical structure of these later. Okay, that's acetylcholine is excitatory because the receptor is a ligand-gated sodium channel. So sodium channel means it lets sodium into the postsynaptic and that means the neuron is depolarizing, which makes it more likely to fire. So to fire means to release an action potential itself. GABA is inhibitory because the receptor is a chloride channel. Chloride, here we actually encounter chloride, makes the cell polarizes the cell, postsynaptic cell, makes it less likely to fire. Other transmitter, dopamine, vasopressins, have G protein-like receptors that can have all sorts of effects. It depends on the signal transduction pathway. So it's different in the visual system than it is in the auditory system. But once it's in the brain, it's the same again. Once it's in the cortex, it's not differentiated anymore. We'll come to that later. Okay, now let's just finish with saying how the signal actually comes from the inner hair cell into the into an action potential in the nerve fiber. The inner hair cell, here, well, two pictures basically showing the same thing. Here a bit more schematically. Potassium from the endolymph, that's the same potassium but in a completely different role. See, the body reuses the parts in different configurations, if you like. The hair cell is not a neuron. Do you know why? It's a sensory cell. What's the difference between a sensory cell and a neuron? The axon and the action potential. You could imagine, if nature was clever, that it would have put an axon on the inner hair cell. It would have saved itself one synapse, but it didn't, because the reason is well, very back in evolution, that these cells are actually doing something completely different. They didn't evolve from neural cells, but they actually evolved from some mechanical cells, probably more muscle cells than anything. So they're not, they have a membrane potential and can do fancy things to the membrane potentials, but they don't have the ability to have an action potential. Only neurons can do that. So we need a translation from the membrane potential in the hair cell into the afferent nerve, the auditory nerve here. And this is exactly what we described now. We know all the ingredients. We have potassium flowing in. It depolarizes the inner hair cell. That depolarization leads to voltage activated calcium channels. It's exactly like in the synapse. In fact, it works like the inner hair cell is a giant synapse. the calcium flows in. The vesicles in the auditory nerve cell is glutamate. That's the one point where glutamate is actually important. It doesn't happen anywhere else, but the glutamate is the one that that is the neurotransmitter that actually is important for the auditory impulse here. That goes into the synaptic cleft, and the afferent neuron, that's the synapse of the postsynaptic neuron, will pick that up. Glutamate is excitatory, so it will start raising its membrane potentials. And the more activity, so the more sound comes in, the more potassium, the more calcium, the more glutamate, the higher the membrane potential. Whew. We still haven't got a spike here, because I said axons. This is an axon, but it will not produce a spike. The spike is actually only produced later on in the cell body, which sits in, you listened in the first week, where's the cell body of the auditory nerves? Spiral ganglion, exactly. If you don't know where that is, go back to the figure, because it sits a few millimeters away from this axon. Okay. Is it just a very large picture to show you something about neural computation? That's my favorite. That's what I make a living with. In reality, they're not green and red. They're blue and yellow. But here, for the purpose, is the green ones are excitatory and the red ones are inhibitory. And this is how it really looks like, or it can be imagined. This is an electron microscopic picture. And you see that it is covered and covered with thousands and thousands of synapses. So each of these branches here actually could come from a different neuron or a different auditory nerve fiber. So in fact, you could imagine this is a cell, the first cell in the auditory system, that is not the auditory nerve, and it actually is connected to 30, 40, 50 auditory nerve fibers coming from the cochlea. And some of these endings are excitatory, some of these endings are inhibitory. And the neuron, you can imagine, they're constantly flickering on and off, and their action potentials coming left, right, and center. And what the neuron does is a very complex computation, averaging all of these pluses and minuses, and calculating. In fact, here, this is where the axon hillock is, because there you have the highest density of these, of the channels and of the synapses. And here it calculates if it is above the threshold or not. It's very complex. Well, it's not a complicated calculation, because in the end it's just adding and subtracting things. There's nothing fancy going on. But because there are thousands, actually, if we talk about the auditory system early on, it's more like hundreds different neurons. If we talk about the cortex, there could be millions of different inputs on each neuron. It becomes more and more complex over time. Okay, and these are the kind of things that postsynaptic neurons can do. This is a postsynaptic neuron, and it has greens, and here's a red ones. Green is excitatory in this case, red is an inhibitory. And imagine you've got one terminal branch of a presynaptic neuron, and it has two inputs, namely one here and one there in time. Check, check. The postsynaptic neuron will not notice anything of that. Well, it will notice that, but it will not reach its threshold, so it won't fire. Okay, there's a subthreshold, no summation. If you have them shorter in time, so not, but then they might add up, because they have a finite length in time, which is in the order of milliseconds. You can add them up, and you might reach threshold if they are fast enough. This is called temporal summation, or coincidence detection. So if several of these things happen in a short time window, there's a coincidence between them, and the postsynaptic neuron will pick that up and fire. More interesting is if you have several of these, E1, E2, and they fire at the same time. That's what's called spatial summation, because you integrate over, so if I say integrate, it doesn't mean mathematically integrate, it means you sum over different inputs from various neurons, and you measure their temporal coherence, in fact. So if they come in a short time window, you will get the threshold, you will get above the threshold. But even if all of that happens, if you have an inhibitory one, you can switch it off. So this is where the minuses come in. These are all the pluses. So far it's only positive, but the inhibitory one is a negative. It actually can stop that. It's actually not a negative in the sense of that you go very far low, but you stop something happening. You stop the axon from firing. That's why actually most of the inhibitory neurotransmitters are released directly at the axon hillock, where the action potential is generated. Okay, this is my slide on neural computation, very basic, because this is all that neurons can do in computational sense. They're not that complicated because they can only work with their membrane potential, and they can either reduce it or increase it, and that as a function of time. Okay, gives me a nice opportunity for a break.